Earlier, we learned that the goal of this course is to test for differences in brain activity between two experimental conditions, incongruent and congruent flanker trials. We expect that brain regions involved in cognitive control, a concept discussed previously, will be more active in the incongruent condition compared to the congruent condition. In order to test this, we will need to first pre-process our data, or clean it up, in other words. If you've ever taken a picture and found problems with it, blurriness, red eye, or faded colors, for example, you can then use an image editor to remove those problems. We do the same thing with the three-dimensional images of the brain taken with the scanner. Each of our pre-processing steps will clean up the images and prepare them for statistical analysis, which we will discuss in a later video. Pre-processing step is done with AFNI commands, which are similar to Unix commands. They typically require at least one input argument, and they usually require you to specify what to call the output of the command. Let's take skull stripping, for example, a common pre-processing step that removes the skull from the brain. The AFNI command to do this is called 3D skull strip. Navigate to the directory sub08 anat and then type 3D skull strip and then press enter. You will see the following message. An error is thrown since the command expects arguments. To see examples of how to use the command, type 3D skull strip -h, a vertical pipe, and then less, which pipes the output of the help file into the less command. When you are in this paging window, type D to go down one page, U to go up one page, and the up and down arrows to go up or down by one line. To search the help file, type a / followed by the string that you want to find. When you are done, press the Q key to exit the paging window. As you can see, the help files are one of AFNI's greatest strengths. The usage of each command is clearly outlined, and the reasons for using different options are explained in detail. Sample commands are given to cover different scenarios. For example, if the skull strip leaves too much skull in the output image, you are encouraged to use an option such as push to edge. From the help file, we also learned that 3D skull strip requires an input flag to specify the anatomical data set that will be stripped. Knowing this, try typing this command, 3D skull strip -input, followed by the name of the anatomical image. After about a minute, a new file called skull strip out plus a rig will be generated. This is the skull stripped anatomical image, which you can look at in the AFNI viewer. Look in all three viewing panes to see how well the skull stripping worked. You'll probably notice a few voxels of cortex being removed in the frontal lobes and some bits of dura mater left around the top and the back of the brain. But overall, the skull stripping did very well. Another way to view the quality of the strip is to load the anatomical image sub08 t1w.nii.gz as an underlay. This is the original anatomical image. And then, as an overlay, you can load the skull-stripped image skull strip out. Once you've loaded both of these, you can alternately view and hide the overlay by clicking anywhere in the viewing windows and then pressing the O key to toggle the overlay being shown and being hidden. Another option is to press the U key to toggle between each image as an underlay. These viewing options will be helpful in any scenario in which you have to examine the data before and after a pre-processing step. Although the skull stripping worked reasonably well, let's see if we can improve it by using the push to edge option that was specified in the help file, which helps avoid removing any parts of the cortex. In general, it's better to err on the side of including small bits of dura mater and other non-brain matter as opposed to removing any parts of the cortex. It's also useful to add a prefix option to label the output as something intelligible. Try the following command: 3D skull strip dash push to edge dash input and then the original anatomical image. Let's call the output anat_ss to specify that it is skull stripped. After a couple of minutes, a new file called anat_ss plus a rig will be generated. Just as we did before, look at the images before and after using the AFNI viewer, and compare it to the skull-stripped image that you generated previously. You can do this by specifying the original anatomical as an underlay, and each of the skull-stripped as overlays. Do you see anything that looks better, worse? Which version would you choose, and why? We could create a text file with all of the pre-processing commands we would use, such as 3D skull strip, and then run them all from the command line. However, this approach is tedious and prone to error. Instead, we'll learn how to use a command called ubersubject.py to create this pre-processing script for us. All that and more in the next video. In the good old days, researchers used to write their own pre-processing scripts from scratch, piecing together different commands to analyze their data. However, since we usually don't know what we're doing, it's better to use a program that will run all of these pre-processing steps for you, AFNI's ubersubject.py. Technically, ubersubject is a graphical user interface that serves as a wrapper. That is, it takes all of the GUI inputs specified by the user and directs it into another wrapper called AFNIproc.py. This command then generates a large script that contains each of the individual AFNI commands needed to run each step of the pre-processing. To see how it works, first navigate to the sub-08 directory. From there, type ubersubject.py, then an ampersand, and press return. For now, look at the first checkbox in the GUI, which is called Analysis Initialization. By default, the data analysis type will be task, as opposed to rest when there's no task for the subject to do. And the domain is volume as opposed to surface, which we will discuss in a later chapter on a program called SUMA. The words in the field after processing blocks, T-shift, align, tie-rack, volreg, and so on, probably don't make sense to you right now. Our goal is to learn about what each of these words mean, what pre-processing step they correspond to, and why we do them. Now let's set up our pre-processing script. In the subject ID field, type sub_08, and in the group ID field, type flanker. Click on the box next to Analysis Initialization and remove the regress block. We'll include this later when we run a general linear model for each subject. Then click on the Browse ANAT button in the Anatomical Datasets section. Navigate to the ANAT directory and select the file sub08 t1w.nii.gz. Select the functional datasets by clicking the Browse Epi button in the Epi Datasets section. Navigate to the func directory, and then hold down Shift and click to select the files sub08 run1 and sub08 run2. We are going to ignore the stimulus timing files and symbolic GLTs sections. We'll come back to them when we do regression. The default values in the expected options field are fine as they are. No TRs will be removed. The volume with the least amount of variability will be used as the reference image for alignment. A smoothing kernel of 4 mm will also be applied to the data. Now check the boxes next to extra align options and extra TLRC options. The default cost function, or algorithm that will align the anatomical and functional images, is fine. However, I recommend checking the box next to Align Use Giant Move. In case the functional and anatomical images are far away from each other, an option called Giant Move will bring them into a closer initial alignment. Lastly, in the extra TLRC options field, choose the template MNI AVG152T1, since MNI templates are becoming the most commonly used templates in the field. When you are done setting up the analysis, you can execute it by clicking from left to right the three icons at the top of the GUI window. The first one looks like a sheet of paper with lines on it. This will generate the AFNiPark2 command that includes everything you specified in the GUI. Click on the icon, and it will return two windows, one listing each of the options that were changed from the defaults and listing each of the inputs, and another showing the code of the AFNiPark2 command. Take a look at it to see how the commands and options listed in the AFNiPark2 command correspond to the options you entered into the UberSubject.py GUI. When you have finished looking over the output windows, close them. Now click on the next icon, a magnifying glass over a sheet of paper. This will execute the AFNiPark2 code listed in the previous window and return any warnings or errors that you should be aware of. We don't have any in this case. You'll also see a couple of lines of code specifying how to run the output of this command, which is a file called proc.sub_08. Close the window and go back to your terminal. From the sub08 directory, notice that there is a new directory structure that has been created. Within the directory subject_results, you will see another folder, group flanker, and below that, a folder called subj.sub08. Within this directory, there are three files you should be aware of. cmd.ap.sub08 is the AFNiPark2 file that was generated by UberSubject.py. Output.cmd.ap.sub08 is the output of AFNiPark2 that generates any warnings or errors that you should be aware of. Proc.sub08 is the raw AFNI commands that will perform the actual preprocessing, as compiled by the AFNiPark.py command. We are now ready to run this script. You can either type the code that was listed in output.cmd, or you can simply press the green icon button at the top of the UberSubject.py GUI. If you do the latter, you will see another window open up that shows each command being run and its corresponding output. Preprocessing will take about 5 minutes, depending on the speed of your computer. When it finishes, you will see the output files from each preprocessing step that was indicated in the processing blocks field. We now turn to reviewing each of these preprocessing outputs, evaluating their quality, and deciding whether there is anything we need to redo. After the script generated by UberSubject.py has completed, navigate to the directory containing the preprocessed data. By default, AFNI will create a new directory tree. From the sub08 directory, you can navigate to the results directory by typing cd subject results group.flanker subj.sub08 sub08.results. This directory contains different versions of the images after each preprocessing step. For example, the files that contain the string pb01 or processing block 01 and the string tshift indicate that these images have been slice time corrected using the 3D tshift command. Now open the AFNI GUI. Click the Underlay button and left-click on the file pb00 sub08 r01.tcat. Then, click on the Graph button next to any of the axial, sagittal, or coronal views to view the time series. Since the initial volumes had already been discarded before the data was uploaded to OpenNeuro, all of the time points are of the same relative intensity. Else, we would see a large spike in the signal intensity at the beginning of the run. Similarly, the PB01 images should be the same as the PB00 images. If you examine the output text from the preprocessing, you will see a message printed during 3D tshift, which states that the data sets are already aligned in time, and that the output data set is just a copy of the input data set. For now, just make sure that the two processing blocks look the same. The next file to look at is the PB02 volreg files, which have been motion corrected, co-registered to the anatomical image, and warped to a standardized space. which in this case was the ENIGMA-U template. If you click on the PbO2 images, the view changes from original view, or the raw data space, to tally rack view, indicating that these images have been normalized. When you view this processing block for other subjects, the basic shape and outline of the images will look nearly identical, since they have all been warped to the same template. Again, check the images and the time course in a few different locations to make sure there are no obvious artifacts. Processing block 3 is smoothing, which averages the signal of the nearby voxels together in order to increase signal and cancel out noise. These images will look more or less blurry as a function of the size of the smoothing kernel that you apply to the data. In this case, a smoothing kernel of 4 mm will blur the data slightly, but not by much. Look at the images to make sure that the blurring looks reasonable for this smoothing kernel size. The last preprocessing step generates scaled images, in which each voxel has a mean signal intensity of 100. This allows us to specify any changes relative to the mean as % signal change. In other words, a value of 101 could be interpreted as a signal change of 1%. Due to the grayscale of the images being more uniform in the brain voxels, as compared to greater variability in the signal outside of the brain, these images will look less defined than the previous images. Nevertheless, you should still be able to see the outline of the brain, and the time series values of the brain voxels should all be close to 100. In addition, AFNI creates a mask that excludes any non-brain voxels. The mask is binary. It will contain ones in the voxels that are determined to be within the skull and zeros outside of the skull. The full mask image is a union of all the individual functional image masks. Voxels with very low signal intensity are not considered brain voxels, and in some cases, voxels are excluded in the orbital frontal cortex, which is susceptible to signal dropout. The mask group image is a more generous mask, conforming more to the outlines of the template brain. Later on, we can use these masks to restrict our analysis to only brain voxels. When viewing the results of the anatomical preprocessing, we will want to make sure that both the skull stripping looks reasonable and that the images were normalized properly. First, open the image ANAT with skull warped. If you have copied the ENIGMA-U image into the A global directory, load it as an overlay image. You can also copy it into the current directory by typing from the terminal cp://abin/mni average 152t1 *. You may notice that while the sagittal view of these two images overlaid looks fine, the axial and coronal views look worse. It seems like the image is shifted to the right. Although it is common to have some variability in normalization, this is more than we would expect. The ANAT-W skull warped image, it should be noted, is the result of a warp being applied to the raw anatomical image. The warp itself was computed by normalizing the skull-stripped anatomical to a template. If that normalization was off somehow, it would have propagated to the other images. To check this, load as an underlay the image ANAT_final. Now we've found the source of the error. Part of the brain on the left has been removed during normalization. But how do we fix this? When you detect an error in the preprocessed images, you should examine the output of your preprocessing script. If you started the script from the ubersubject.py GUI, the output will be printed to the preprocessing command window. A copy of the text will also be stored in a file called output.proc followed by the name of the subject ID, which is located one directory above the preprocessed data. This text will contain both warnings and errors. Errors indicate that either a file is missing or that a command was not able to run successfully. Usually the script will exit after an error is encountered. Warnings, on the other hand, point out something that may be a problem. An example of a warning is the data set already aligned in time notification that we received during slice timing correction. Another warning related to our current problem occurred during the normalization step. This can be found slightly after the halfway point of the output after the @AutoTLRC command. Apparently, the centers of the anatomical and template images are very far apart. The output says that if parts of the original anatomy gets cropped, which is our current problem, try adding the option init xform autocenter to your AutoTLRC command. We can do so by navigating to one directory above the preprocessing directory, removing that preprocessing directory, and then editing the file proc.sub08 to include the string init xform autocenter after the AutoTLRC command, which should be in line 119 of your proc file. Here I'm opening the file in Text Wrangler. We then locate the AutoTLRC command and we add the additional option init xform autocenter. Save the file and rerun it by going to the terminal and typing tcsh proc.sub_08. As before, wait a few minutes for it to finish, and then navigate into the preprocessing directory and load the same set of images as before. You should now see that the problem is fixed. Now that you've preprocessed the data, we can fit a statistical model and estimate the amount of brain activity in response to our experimental conditions, which we will cover in the next video.